before we proceed to SOP, I'll tell you why golden apple snail are the major pests in our problem, and why wild passion fruit can can have a can act as a mollusticidal spray to this problem. Nowadays, golden apple snail, Pumachea caniculata, are one of the most major pests in rice production. Originally found through the Amazon River basin, it has now spread to Hawaii, Southeast Asia, and has been recently reported in Florida. They were introduced to from South Africa in effectiveness of wild passion fruit, otot-otot, Passiflora edulis, fruit and leaf extracts, and golden apple snail, Pumachea caniculata, as a mollusticidal spray. And these are the researchers: Gardoki Flores May D, Yuson KCC, Benaflor Jola Isaac B. So, nowadays, golden apple snail are one of the most major pest problems in rice population. Originally found throughout the Amazon River basin, it has now spread to Hawaii, Southeast Asia, and has been reported in Florida. They were introduced to Asia from South America in 1980s as potential food for people, but unfortunately become a major pest of rice. Golden apple snail eat young and emerging plants. They cut the stem at the base, destroying the whole plant. So the only cure for this problem is the wild passion fruit. And and wild passion fruit or otot-otot belongs to Passifloracea family, with about 500 species distributed in warm temperature and tropical regions. Studies have known antibacterial, anxiolytic, anti-inflammatory, and lipid benefit activities. The researchers also found that wild passion fruit contains component that is worthy of being mollusticidal spray, such as saponins, tannins, alkaloids, phenolic compounds, flavonoids, and terpenoid. So our SOP, or statement of the problem: this study will aim to test the mollusticidal ability of wild passion fruit, otot-otot, Passiflora edulis, fruit and leaf extracts, and golden apple snails, Pumachea caniculata. Moreover, it will aim to answer the following questions. Question number 1. How will the researchers make a stable mollusticidal spray using wild passion fruit, otot-otot, Passiflora edulis, fruit and leaf extracts, and distilled water? Question number 2. What is the procedure in extracting the active components in the fruit and leaf extracts of wild passion fruit, otot-otot, Passiflora edulis? Question number 3. Is there any significant difference in the time it takes to kill golden apple snail between the leaf and fruit extracts of wild passion fruit as a spray and other organic mollusticide? So this is our hypothesis. Null hypothesis: there is no significant difference in time it takes to kill golden apple snails between the leaf and fruit extracts of wild passion fruit as a spray and other organic mollusticide. Our alternative hypothesis: there is a significant difference in time it takes to kill golden apple snails between the leaf and fruit extracts of wild passion fruit as a spray and other organic mollusticide. And these are conceptual framework. Input: wild passion fruit, otot-otot, Passiflora edulis, fruit and leaf extracts. First, 100% concentration of fruit and leaf extracts of wild passion fruit. And then second, 80% concentration of fruit and leaf extracts of wild passion fruit and 20% distilled water. The third, 60% concentration of fruit and leaf extracts of wild passion fruit and 40% distilled water. Moderating variable: mortality rate of the golden apple snails, Pumacea caniclata, and the output, quality of molluscide spray.
Chapter 2, Research Methodology. Research Design. The research study will use an experimental design to see if the treatment or idea stated will make a difference. There will be a control group and a treatment group. One group will get the treatment and the other group will not get the treatment. There will be a post and pre-test for both groups as followed in a traditional experimental design to know the difference or effectiveness of the treatment or idea. Research Local. The ingredients of the multicidal spray will be obtained from two different places. The wild passion fruit, Passiflora edulis, will be taken from the bushes of the nearest suburban and rural areas, while the distilled drinking water will be bought from the nearest convenience store of the makeshift laboratory. The researchers will buy the solvents needed in extracting the needed active components from the fruit and leaf extracts from one of the researcher's father's friend who is a chemist in a company. The before-mentioned products will be made at one of the researcher's house. The student researcher's house will be equipped with all the materials needed to proceed with the experimentation. The researcher's makeshift laboratory will be the kitchen. The sample golden apple snails from Pumacea caniclata in the experimentation will be from the nearest rice fields. The application of the multicidal sprays will also be at one of the researchers' house. Research sample. The researchers will randomly choose a crop with golden apple snails, Pumacea caniclata, specifically a crop that has 3 golden apple snails. The 3 golden apple snails will be sprayed with the treatment, the positive control, which is the organic commercial multicide, and the negative control, which is the water, respectively. The 3 golden apple snails will serve as samples. For the sampling technique, the researchers will be using convenience sampling. With convenience sampling, the samples will be selected because they are accessible to the researchers. Samples will be chosen simply because they are easy to obtain, according to Makadagal et al. 2015. Statistical treatment. The study will make use of ANOVA, or analysis of variance, which will determine the significant differences among the 2 or more variances. The researchers will use ANOVA because they have 3 groups to compare in specific. Sources of data and data gathering. The data collection method that the researchers will use is the experimental method. It is a method that you use to compare a treatment group with positive and negative control groups. The information gathered in the study is from the experiment with manipulated conditions.
Research procedure. Preparation of the multicidal spray. The researchers will supply the main ingredient for the research, which is the wild passion fruit, utot utot, scientific name, Passiflora edulis. Fruit and leaf. The fruit and leaf of wild passion fruit, utot utot, Passiflora edulis, will be taken from the bushes of the nearest suburban and rural areas. Next, the researchers will do the extracting of the active components from the fruit and leaf of wild passion fruit, utot utot, Passiflora edulis. They will use different methods of extracting each active component. The researchers will use methyl for the extraction of the tannins and flavonoids, ethyl for the extraction of terpenoid and phenolic compounds, ethanol and others for the extraction of alkaloids and saponins. The other materials, such as the 300 ml distilled water, the steering rod, the spray bottles, the sterilized drug, lid, marker, and plastic gloves will be found at the student researcher's house. On getting the wild passion fruit, fruit and leaf extract with the active components, the researchers will use the different extracting methods as mentioned before. Combine the 300 ml distilled water. We use distilled water and not tap water for there will be no germs and no bacteria. And wild passion fruit and leaf extracts into a sterile jar. It's supposed to be sterilized to, again, be free from the germs and the bacteria. And stir it with a stirring rod. Cover and set aside for 24 hours, preferably in a warm place. Strain using the strainer and pour it into the spray bottle. Lastly, clearly label the bottle. Ethical considerations and proper waste disposal. The researchers will make sure that the rice crops won't get affected by the product they sprayed on the golden apple snails, scientific name Pomachea caniculata. The researchers will also ensure their safety in doing the experiment. On the other hand, proper waste disposal will be observed. The waste from the fruit will be properly disposed by putting all of it in a plastic cellophane and labeling it with fruit waste. For the golden apple snails, Pomachea caniculata, if they are successfully eliminated, the researchers will handpick them and of course wearing plastic gloves to ensure the safety. After so, they will dispose them by gathering them and also putting it in a plastic cellophane also, labeled with animal waste, and putting all the waste in their corresponding wheelie bins. Flowchart. First, the researchers will gather all the needed materials. Next, the researchers will begin the extracting of the active components in the fruit and leaf extract of the wild passion fruit. The researchers will make the molluscicidal spray. The researchers will start the application of molluscicidal spray on the samples, the golden apple snails. They will start the statistical treatment using the ANOVA. The researchers, will begin the analyzation of results. The researchers will formulate a conclusion. And then lastly, the researchers will utilize their product and communicate their results.